Today I'm going to be teaching you guys about 8.2, cellular respiration, by Max Silver. Before I talk about cell respiration, it consists of several different interlinked metabolic pathways, and they are glycolysis, link reaction, Krebs cycle, electron transport change, and chemiosmosis. Glycolysis is the splitting of glucose into pyruvate. It uses 2 ATP and it produces 4 ATP. Its net yield is plus 2 ATP. Glycolysis occurs in the cytoplasm. A hexose sugar, for example glucose, is phosphorylated using ATP. The hexose phosphate is then split into 2 triose phosphates. Oxidation occurs, removing hydrogen. Then the hydrogen is used to reduce NAD to NADH. 4 ATP are produced, resulting in a net gain of plus 2 ATP. Finally, 2 pyruvate molecules are produced at the end of glycolysis. Next is the link reaction. In link reaction, the pyruvate from glycolysis enters the mitochondrion matrix. Then the enzymes remove 1 carbon dioxide and hydrogen from the pyruvate. The hydrogen is accepted by the NAD to form NADH. A removal of hydrogen is called oxidation. A removal of carbon dioxide is called decarboxylation. The link reaction is therefore oxidative decarbolization. The product is an acetyl group which reacts with coenzyme A. Acetyl coenzyme A enters the Krebs cycle. Now that we are in the Krebs cycle, let me take you through it. Acetyl coenzyme A enters the Krebs cycle. Then, the acetyl group 2C joins a 4C sugar to form a 6C sugar, because 2 plus 4 is 6. Then the oxidative decarbolization of the 6C sugar to a 5C compound produces CO2. Oxidative decarbolization of the 5C compound to a 4C compound produces CO2 as well. The process is oxidative as NAD and FAD are reduced by the addition of hydrogen. 2 CO2s are produced per molecule of pyruvate, along with 3 NADH plus H plus and 1 FADH2 per molecule of pyruvate. 1 ATP is produced by substrate level phosphorylation per molecule of pyruvate. And finally, NADH and FADH2 provide electrons to the electron transport chain. Next is the electron transport chain. Choo-choo. The electron transport chain is situated on the inner mitochondrial membrane. Hydrogen is transferred to the electron transport chain by hydrogen carriers, which are NADH and FADH2. The hydrogen carriers release electrons, which are transferred between carriers. This releases energy, which is used to pump H plus ions across the inner membrane. The plus H ions to accumulate in the inner membrane space, creating a concentration gradient. These ions return to the matrix through ATP synthase. Down the electrochemical concentration gradient, this produces ATP by chemiosmosis. This is the final step. The final step is chemiosmosis. Chemiosmosis is the diffusion of ions across a semi-permeable membrane through a carrier protein. In this case, the ions are hydrogen protons and the carrier is ATP synthase. The flow of the hydrogen proton through an ATP synthase generates ATP. And this was section 8.2, cell respiration. Thank you.